Welcome to Chapter 31, Conduction System of the Heart and Electrocardiography. In this lesson, we'll explore how the heart's electrical system coordinates every heartbeat with remarkable precision. The heart is not just a muscle, it's an electrically driven pump that generates its own rhythmic impulses. Through this system, the atria and ventricles contract in perfect sequence to maintain efficient blood flow. By studying the conduction system and interpreting the electrocardiogram, or ECG, we can visualize these electrical events and identify cardiac abnormalities such as arrhythmias or heart block. Understanding this connection between structure, function, and rhythm forms the foundation for diagnosing and managing cardiovascular conditions. This image introduces the study of the heart's electrical system, captured here as an electrocardiogram, or ECG. Each small wave on this tracing represents the coordinated depolarization and repolarization of the heart's chambers, the electrical events that trigger every heartbeat. The ECG allows us to visualize the invisible language of the heart's conduction system, revealing its rhythm, rate, and regularity. Through this chapter, we'll explore how specialized cardiac cells generate impulses, how those signals spread through the myocardium, and how to interpret the patterns they produce. Understanding this electrical blueprint is essential to diagnosing cardiac abnormalities and connecting electrical activity to mechanical function in the living heart. The intrinsic conduction system of the heart allows it to beat independently of direct nervous control. It begins with the sinoatrial node, or SA node, often called the heart's natural pacemaker. Located in the right atrium, it generates impulses about 75 times per minute. These impulses travel through internodal pathways to the atrioventricular node, or AV node, which briefly delays the signal. This pause ensures the atria contract and empty their blood before the ventricles contract. From the AV node, impulses descend through the bundle of His, split into right and left bundle branches, and spread through Purkinje fibers, causing the ventricles to contract from the apex upward. This organized flow guarantees an efficient heartbeat. Let's review the pathway of electrical conduction through the heart. The process starts at the SA node, spreads through the atria, and reaches the AV node, where a short delay occurs. Then, the signal travels down the bundle of His, continuing into the right and left bundle branches within the interventricular septum. Finally, it reaches the Purkinje fibers, which distribute the impulse throughout the ventricular myocardium. The result is a synchronized contraction pattern, atria first, ventricles second, that ensures efficient pumping of blood. The coordination of these depolarization waves underlies the rhythmic heartbeat and can be visualized on an electrocardiogram. An electrocardiogram, or ECG, records the electrical activity of the heart as it contracts and relaxes. Each wave on the tracing represents a specific event. The P wave reflects atrial depolarization, initiating atrial contraction. The QRS complex corresponds to ventricular depolarization and the beginning of ventricular contraction. The T wave represents ventricular repolarization, or recovery, as the ventricles prepare for the next beat. Together, these waves form a complete cardiac cycle that repeats with each heartbeat. The ECG provides valuable information about heart rhythm, conduction speed, and the health of the myocardium. In a normal ECG tracing, the timing and duration of each wave are critical indicators of cardiac function. The PR interval extends from the start of the P wave to the beginning of the QRS complex, representing the time it takes for the electrical impulse to travel from the atria to the ventricles, typically between 0.12 and 0.20 seconds. The QRS duration is short, reflecting rapid depolarization of both ventricles. The QT interval measures the time required for ventricular depolarization and repolarization, which corresponds to the heart's electrical refractory period. By analyzing these measurements, clinicians can detect conduction delays or irregularities that may signal underlying disease. Alterations in ECG intervals reveal important clinical conditions. A prolonged PR interval indicates delayed conduction through the AV node, often classified as a first-degree heart block. As the block worsens, second and third-degree heart blocks can occur, where atrial and ventricular contractions become dissociated. A shortened PR interval may suggest an abnormal conduction pathway, such as in pre-excitation syndromes. The QT interval represents the duration of ventricular action potentials, its prolongation can predispose the heart to life-threatening arrhythmias. These measurements are more than just lines on paper, they reflect the heart's electrical integrity and the timing of its contractions. On this slide, we focus on the Q and S waves, both critical parts of the QRS complex on an ECG. The Q wave represents the initial depolarization of the interventricular septum, where the impulse travels from the left to the right side of the septal wall. This marks the beginning of ventricular activation. The S wave follows as the final phase of depolarization, signaling the spread of the impulse through the thick walls of the ventricles. Because the left ventricle has a much greater muscle mass, it contains more Purkinje fibers and therefore generates a stronger electrical signal. Together, these waves complete the QRS complex, reflecting the rapid, synchronized depolarization that initiates ventricular contraction and drives the main pumping action of the heart. The PR interval measures the time from the beginning of atrial depolarization to the onset of ventricular depolarization, normally lasting between 0.12 and 0.20 seconds. It represents the delay as the impulse travels through the atria, AV node, and into the ventricles, allowing the chambers to contract in proper sequence. When the PR interval is prolonged, it signals a conduction delay, known as a heart block. In a first-degree heart block, the signal is simply slowed. In a second-degree block, some impulses fail to reach the ventricles, creating skipped beats and often requiring treatment. A third-degree, or complete, heart block means that the atria and ventricles beat independently, with no communication between them. Conversely, a shortened PR interval indicates that the impulse bypasses the SA node or takes an abnormal accessory pathway. Recognizing these changes is essential for diagnosing conduction abnormalities on an ECG. The QT interval represents the total duration of ventricular electrical activity, from the start of depolarization through the end of repolarization. It corresponds to the refractory period, a critical window during which cardiac muscle cells cannot generate another action potential. During this time, the ventricles are contracting and must complete their cycle before the next impulse begins. The absolute refractory period occurs first, no new action potential can form because the voltage-gated sodium channels are closed. This mechanism prevents the heart from entering tetany, or continuous contraction. Following this phase, the relative refractory period allows a second action potential to occur, but only if the stimulus is strong enough. Together, these phases ensure rhythmic, controlled heartbeats and protect the myocardium from erratic electrical activity that could disrupt normal rhythm. This table summarizes the normal duration and boundaries of each ECG component. For example, the P wave reflects atrial depolarization and typically lasts less than 0.12 seconds. The PR interval should not exceed 0.20 seconds, and the QRS complex should remain narrow, under 0.10 seconds, indicating efficient ventricular depolarization. The QT interval, which varies with heart rate, generally falls below 0.44 seconds. These reference values help healthcare professionals interpret ECGs accurately and identify abnormalities in electrical timing, conduction velocity, or myocardial recovery. Abnormal ECGs can provide early clues about cardiac dysfunction. Sinus tachycardia shows a normal rhythm with a rapid rate, often due to stress or fever. Bradycardia presents a slow but regular rhythm, sometimes seen in well-trained athletes or in SA node disease. Atrial fibrillation produces irregular P waves and chaotic atrial activity, while ventricular fibrillation displays disorganized electrical signals and no effective contraction, an immediate medical emergency. Heart blocks, premature beats, and ischemic changes all leave distinct signatures on an ECG. Recognizing these patterns allows clinicians to diagnose conditions quickly and guide treatment decisions. This figure connects electrical activity with mechanical contraction. As the SA node fires, atrial depolarization produces the P wave, triggering atrial contraction. The impulse travels through the AV node and down the bundle branches, generating the QRS complex, which represents ventricular depolarization and the start of systole. Following this, ventricular repolarization occurs, forming the T wave, as the ventricles relax and refill. Depolarization always precedes contraction, while repolarization prepares the myocardium for the next beat. Disruptions in this pattern can cause arrhythmias, leading to reduced cardiac output and clinical instability. This figure compares 6 fundamental cardiac rhythms that can be identified on an electrocardiogram. The normal sinus rhythm shows evenly spaced complexes with a rate between 60 and 100 beats per minute, representing a healthy and coordinated heartbeat. In arrhythmia, all waveforms appear normal, but the rhythm becomes irregular due to disruptions in the conduction pathway. Bradycardia presents a normal rhythm with a rate below 60 beats per minute, often seen in athletes or during rest, while tachycardia shows a rate above 100 beats per minute, typically caused by stress, fever, or cardiac disease. Atrial fibrillation is marked by chaotic atrial activity, producing an irregular baseline and inconsistent ventricular response. Finally, ventricular fibrillation represents complete electrical disorganization, rapid, wide, irregular complexes with no coordinated contraction, an emergency that requires immediate defibrillation. Recognizing these patterns is essential for interpreting ECGs and assessing cardiac function in real time. Cardiac output, abbreviated CO, is a measure of the heart's efficiency. It represents the volume of blood pumped by each ventricle per minute and is calculated using the formula CO equals stroke volume times heart rate. Stroke volume, or SV, depends on the amount of blood entering the ventricle and the force of contraction, while heart rate HR is the number of beats per minute. Any change in these two factors, such as increased rate during exercise or reduced volume during dehydration, directly affects cardiac output. This relationship demonstrates how electrical control translates into mechanical performance. Starling's law of the heart states that the greater the volume of blood filling the ventricles during diastole, the stronger the contraction during systole. This is due to optimal overlap of actin and myosin filaments within cardiac muscle cells, which enhances contractile force. Essentially, the heart automatically adjusts its pumping strength in response to venous return. This intrinsic regulation ensures balance between the amount of blood entering and leaving the heart, maintaining efficient circulation without overstretching or underfilling the chambers. This diagram illustrates the anatomical locations of the heart valves and the optimal sites for auscultation, where we listen for their sounds using a stethoscope. Although the valves lie close together deep within the chest, each sound is best heard at a distinct surface landmark. The aortic valve is auscultated at the second intercostal space to the right of the sternum, while the pulmonic valve is heard at the second intercostal space to the left. The tricuspid valve is best heard at the lower left sternal border near the fifth intercostal space, and the mitral valve is auscultated at the cardiac apex, typically at the fifth intercostal space along the midclavicular line. Listening at these specific sites allows clinicians to distinguish between normal heart sounds, S1 and S2, and abnormal murmurs that may indicate valve disease or dysfunction.